For today, our first presenter will be Shree Ishwarya K. She is a content writer at Scioverleaf and she is a bachelor student doing B.Tech Biotechnology. Kindly give a kudos to her and Shree Ishwarya, you can present now.
Thank you. So, good evening, all. Today, we are going to speak about a topic which is going to edit your genome through some interfacial forces. This tool is called as a vector. Vector is nothing but it is a tool which is going to reorganize your genome into something you wanted, something you like. Okay, so before going to the topic, I'm Shree Ishwarya, studying in Rajalakshmi Engineering College and a content writer in Scioverleaf. So coming upon to our topic. So vector has an expansion in it. It is a viscoelastic chromatin, threading, and organization tool. So this tool is going to manipulate your chromatin and organize it according to you or your desired organization. So this is what basically vectors deals with. In this tool, we can also organize the genome as well as you can study the genome materials or the material inside the genome. Okay. So basically here we deal with a chromatin material. All you would have know what is meant by chromatin. It is nothing but a thread-like structure that consists of your DNA and protein, which is going to coil it and make the X-shaped chromosomes. So this is called as chromatin material. So how does this chromatin material alter your genome? So whenever this is containing your DNA and proteins, right? So whenever this chromatin is going under some changes or some type of stress like cellular changes or extracellular changes, this is going to influence your replication, transcription, and the repair mechanisms of the DNA. So that is why it is very important to study this chromatin. So particularly this chromatin molecules mechanical property, not only the chemical property and also the mechanical property and the environment where the chromatin is present influence your gene regulation and your cell differentiation. Basically, it is division of cells. Okay, so that is why it is important to study your chromatin. So this chromatin is two types. You have euchromatin and heterochromatin. Euchromatin is nothing but a loosely packed chromatin. You can see here like a loosely packed chromatin thread runs over here, whereas in heterochromatin it is tightly packed chromatin. So upon some modifications like DNA methylation or histone methylation, this euchromatin is going to become heterochromatin. So this process is a reversible process. So that is how your cell division is happening. Okay, so I hope you have an idea about chromatin now. So coming, so this is also the conversion of euchromatin and heterochromatin due to some changes in your cell environment. So this is what going to happen basically when your chromatin is undergoing some type of stresses or chemical changes inside it. So either it is going to convert as heterochromatin due to the changes or due to euchromatin. So this euchromatin here, right? This is a loosely packed chromatin, which is exceptionally present in the cancer cells. Whereas in healthy cells, the chromatin is tightly packed. Okay, so remember that in cancer patients, the chromatin is loosely packed, whereas in healthy people, the chromatin is heterochromatin. Okay, so coming upon to a question here. So do you think I just told that this vector can study your mechanical properties or state of matter of your chromatin? So what do you think the state of chromatin here is? Whether it is elastic compound or it is a solid or a liquid? Just comment in the box or unmute and speak. Any guesses or we could have moved it? Hope there is no guess or something. Okay. Okay, I'll answer this question in the end of the session or the upcoming slides. Okay. So before going into our vector system, so what are the traditional method that is used before before for studying our chromatin and their limitations, because then that's when you understand why it is very important to use vector here. So first is a static measurements. This static measurements is where you're taking a cell and it is in the fixed state, and this cell is dead by now when you are analyzing it, and you are analyzing only the chromatin at a point of time. This chromatin changes dynamically. Time to time, this chromatin is changes. Here in static measurements, you are just analyzing a chromatin in particular time. Therefore, in this method, there is no real-time analysis, and this is limited to only few cells. We can't analyze more number of cells here. So, for example, here is high throughput chromosome confirmation capture. This is commonly called as HIC technique. So other than the static method, we have antibody-based technique. Here we use the antibody like antibody particles that is going to attach to your chromatin compounds and then detect the component present in it. So here, what is the limitation is we have to have the antibody which is specific to your chromatin. Other antibodies cannot bind to it. So that is what the limitations here. The example for this method is chromatin immunoprecipitation. This is used to study the modification, that is the euchromatin to heterochromatin modifications in the system, in your biological system. So this method, we need an antibody compulsory. So this is what the limitations here. Whereas in image-based technique, you're going to have your microscope and then the image of your chromatin. So here the microscope cost and the image resolution has plays a very impact on your uh, study, like studying the chromatin molecules. So that is what here the main limitations here. Whereas in vector, the equipment cost is much less. And here is some other technique that is traditionally used, and there are limitations here. So, for example, in HIV technique, we can't analyze the chromatin in real time. In ATAC sequencing technique, there is no mechanical property. That is, you can't measure the state of chromatins here. So that is what some limitations. And in fish, you again need a fixed cell, and we can't track the live cell. So how does this vector overcome these limitations here? So in vector, you can analyze a live cell and you can also check the mechanical properties of chromatin, because these mechanical properties affect your genome organization. And this detect the chromatin materials in precision. And this does not need any antibody like previously discussed in antibody-based technique. And this is very versatile technique also. So coming upon the basic science, how does this researchers got the idea behind this vector is because of the biomolecular condensate. These condensate or particles that is present inside your cell scattered somewhere. Okay, so this condensate come together and form a membraneless structures due to the liquid-liquid separation. So this condensate inside your body is called as biomolecular condensate. So what does this play a role? So this biocondensate go attached to your chromatin material or any type of biological compound inside your cell. These go and attach to them and alter their organization. So this plays as a key role or a key player in organizing your chromatin naturally. So this is the idea. This is the basic idea behind the vector. So we can see these examples of biomolecular condensate here. So, for example, we have MYC condensate. This is antibody condensate, and a fusion protein condensate. This is an example of two proteins combined together and form a condensate. Whereas here in vector, we're going to deal with nucleolus associated condensate, because these condensates are present inside your nucleus and going to alter the organization of your genetic genome. And coming upon to the vector, here in vector system, we can't use the biological condensate or a natural condensate that is present inside your body, because we can't find where is the condensates, right? So therefore, we are going with a synthetic condensate. These are uh, organized or these are done in the laboratory and then introduced into your body for chromatin repositioning. And for this also measures the mechanical properties of vector and also repositioning of the DNA. So using vector, we can reposition your DNA as well as you can measure the property or analyze the chromatin. So what are the key features here is this is versatile. So in whatever chromatin material or whatever material you can analyze it, and it is scalable. That is, it is not cost very cost or high in cost. So it is very less expensive compared to other methods. So the mechanism. So what is here mechanism is you have a synthetic condensate. You can see this green color uh, bubble-like structure is your condensate, which has two proteins. One is SSBB protein and GFP protein. This is a biomolecule. This is a condensate which we are going to insert into your body, and we have an adhesion molecule. Adhesion molecule is also the TRF1 protein and your chromatin sequence. Okay, these two condensate molecule and adhesion molecule make the correlate system. So this system is going to relocate your genome. So whenever this condensate molecule get attached to the chromatin, you can see in the second picture, like in activation and deactivation, right? Whenever the condensate, that is a green color bubble, going to attach with the chromatin, these two chromatin loci are coming together due to the capillary force that is exerting due to condensate molecule. So upon dissolving of this condensate, because this is synthetic molecule, right? So that it gets dissolved after some times. So this biomolecular or synthetic condensate get dissolved and shrinks. Therefore, the two chromatin nuclei, the purple color, comes together. So therefore, coming together, we are saying that the chromatin is relocated using vector. And the application of genome organization. So the main application behind vector is genome organization. As I said, you can arrange your genome. So they are taking the two types of genome organization. One is telomeric end, the other one is non-telomeric end. Telomeric end is nothing but the telomeric ends are the ends of DNA, last few base pairs of DNA, which are called as telomeric end. Whereas the first or the other than last, the other nucleotide bases, all are non-telomeric ends. So end bases are telomeric and non-end bases are non-telomeric ends. So whenever they want to reorganize the telomeric end, the it is very possible. Therefore, it is very possible, and they had made a successful step towards it organizing the telomeric end. Whereas in non-telomeric end, the molecules get is not coming together. You can see clearly here. This condensate molecule and the chromatin loci cannot attach to it. Okay, so cannot attach to it. Due to that, the forces that the capillary forces that is acting on the chromatin loci is not bringing those two chromatin loci nearer. So therefore, they concluded that this vector mechanism is limited to only telomeric end, and uh, it is not feasible for doing non-telomeric end. However, they are going to measure. They are taking steps to alter the non-telomeric ends also with this vector molecule. So this is what the basic idea between genome organization. And as I said, using vector, we can also measure the properties of chromatin, right? So how are we going to measure that? So here we are applying the force, and an artificial force is applied to the chromatin and then released. So scientists or researchers have observed two phenomena. One is elastic recoil, the other one is viscous recoil. Elastic recoil is nothing but they are applying the force and releasing the force. And we can see the complete recoil of the chromatin. So whenever the chromatin is completely recoiling or completely going to their original position, is called as elastic recoil. For example, you can remember this as a rubber band. Whenever you are stretching and leaving it, it goes to its original position. It is not staying in the stretched position itself, right? So therefore, this phenomena is called as elastic recoil. That is, it is going back to its original position. Whereas in viscous recoil, it has an incomplete recoil. Whenever the force is released, the chromatin material is not going to its original position. So that is an incomplete recoil in most of the cases. Therefore, the scientist has the researchers concluded that chromatin is a viscoelastic liquid particle, not a solid or not an elastic. It is a viscoelastic liquid because it is not like it is neither not recoiling or completely recoiling. It is incompletely recoiled. So it is an elastic material, but a viscous also. So therefore, it is viscoelastic liquid. So have you got the answer that for the question that I've asked before? So what is the material state of chromatin? It is viscoelastic liquid. So coming to the epigenetics and disease research applications of vector, what is mean by epigenetics? Epigenetic is nothing but, as I said, euchromatin is shifting on to the heterochromatin by using a protein called as histone. So this blue color and yellow color parts are the histone protein that is going to wrap your DNA and convert the euchromatin into heterochromatin. Okay, so you can see DNA with the help of histone protein, it is going to chromatin and then to a chromosome, the X-like structures. Okay, so this is what the epigenetics, the modification of DNA is called as epigenetics. So this effects can be measured using the vector. For example, in cancer cell, as I said, they have an specific mutations or changes in the telomeric ends alone. Therefore, the telomeric ends of telomeric ends are euchromatin or converting the heterochromatin to the euchromatin region. Okay, so this is what happening basically in the cancer cell. For eliminating or converting the euchromatin back to heterochromatin, this vector technique is used. You can clearly see the difference between healthy and cancer cell using vector. For example, the blue color, the PODH gene is in tumor suppressor gene in the healthy individual, whereas in cancer the same gene is present in oncogene, but here the DNA particles are loosely packed. So that is where the vector comes into and reposition the gene and the chromatin. So basically, it is treating the disease also. So thank you for patiently listening to it. I hope everyone have understand the topic.